break, we were looking at different kinds of proteins, and we looked at proteins in general, and then we looked at enzymes in particular. And now we're going to look at the process of protein synthesis, which is how proteins are made. There is what we call a law in biology, and that is the law called the central dogma. In all living organisms, whether you are a very tiny little bacterium, or whether you are a human, and all of the organisms in between, all of your metabolism follows the central dogma, and that is DNA makes RNA, and that process is called transcription, and RNA makes protein, and that process is called translation. And here we can see that we have the double helix molecule, or the double-stranded DNA, and we're going to make RNA, that is transcription. We're then going to take this RNA and we're going to make a protein. So let's see how this works. We're going to start off with the process of transcription. Now that word comes from the word scribe. And a scribe in ancient times was a person that sat and copied books out precisely without making any mistakes. A scribe was a very important person because great amounts of very important information was entrusted to them for them to copy out books, because no printing presses existed at that time. And that is the role that RNA plays. It has to copy the DNA. In transcription, we're going to see that the RNA that is formed is a special type of RNA called messenger RNA. So let's have a look at this process called transcription. It's going to copy the nucleic acid code from DNA to messenger RNA. So we have a little diagram here of a stretch of DNA. Now remember that DNA is precious. It is carrying your whole complement of all your genes. It can't move out into the cytoplasm to make proteins by itself. Just like very important documents are not allowed to leave archives or libraries. You can make photocopies of them or photographs of them, but you can't take the original one away. In exactly the same way, the DNA stays inside the cell nucleus, and a copy of the gene that we want is made. So along that stretch of DNA, let's imagine that we have this one gene, and this gene X codes for a particular protein that we need in the cell. At that particular site of that gene, not down the rest of the DNA strand, just where that gene is, the DNA unwinds and it unzips the bonds that join together the various different nucleotides break. And RNA copies that original DNA template. If we have a look at what happens afterwards, we see that the DNA at the site of that gene rolls up, re-zips, and it's perfect. It's just been copied, that's all. It would be like you want to borrow your aunt's recipe for chocolate cake. She's not going to trust you to take her whole recipe book home with you for you to make the chocolate cake at home, but you can go and sit in her home and you can copy out the one recipe for chocolate cake. That is exactly what the mRNA has done. It's copied one gene, and that one gene codes for one protein that needs to be made. And if we look at the structure of that messenger RNA, we see that it looks very similar to the DNA, except for it is a single strand, not a double strand, and the nitrogenous bases are made up of adenine, guanine, cytosine, but uracil replaces thymine. So we have uracil here instead of thymine. It still has the ribose sugar phosphate backbone. And what is very important here is the order of the bases. the order was copied directly from the DNA in a complementary manner. So if our DNA order went adenine, thymine, cytosine, cytosine, adenine, for example, the messenger RNA would be uracil, adenine, guanine, guanine, uracil. We know that adenine and thymine are usually complementary, but because we have no thymine on messenger RNA, we complementary base pair it to uracil. Thymine is going to bond with adenine or produce its complementary base pair of adenine and guanine and cytosine. And so that original gene has been copied. And the DNA is a huge molecule, but the messenger RNA that has copied just the one gene is much, much smaller. And so it can very simply move out of the nucleus into the cytoplasm through one of the nuclear pores. Right, so we've just looked at the process of transcription, copying the nucleic acid code. After the break, we're going to look at translation, which is what happens inside the cytoplasm. So come back after the break.